before kind of explaining our work, it's important to explain the context for this. And really, this work began with a new understanding about myelin and its role over the lifespan in the brain. And specifically, we used to think that myelin is really formed during the development of the brain, and then it's just kind of unchanged, throughout most of our lives. And myelin, is an insulating substance that coats the projections that come off of neurons or the brain cells in our brains, and it makes signal transmission in the brain more efficient and more coordinated. So we used to think that myelin, is formed on neurons early in brain development, and then is unchanging after that. However, within the last decade, work by one of my mentors, Dr. Michelle Mange, and other labs around the world has really demonstrated that actually myelin is more dynamic than we used to think. Specifically, neuronal activity can influence the structure of myelin, and new myelin can be formed or myelin can be remodeled in response to different patterns of neuronal activity. And it turns out that in the healthy brain, this myelin plasticity, or activity regulated myelin plasticity, is an important part of the adaptive response to experience, such as learning or honing a motor skill, for example. So that set the stage for my work. And in my case, I'm a physician who cares for children with epilepsy. And of course, epilepsy is a disease that is defined by recurrent patterns of pathological neuronal activity. And so I wondered how pathological patterns of neuronal activity, such as seizures, might influence the structure of myelin, and how those changes in myelin, if they were to occur, might subsequently influence the behavior of the brain. So that was really the question, or the questions that we were seeking to answer in our study. So what we did is we decided to focus on one type of epilepsy. Of course, there are many different types of epilepsy that, they have in common the feature of recurrent seizures, but they are very different diseases. So we focused on one type of epilepsy, which is generalized epilepsy with absence seizures. And we were looking in this initial study in a couple of different rodent models of generalized epilepsy with absence seizures. And both were genetic models that spontaneously have this type of seizure. And in case the viewers aren't, familiar with absence seizures, these are just brief but very frequent seizures that are outwardly manifest by behavioral arrests or sort of freezing and staring spells. And on EEG, they have a generalized spike wave discharge pattern. And one really interesting feature of these animal models is that they have characteristic patterns of seizure onset followed by progression. So the seizures start and then, but they're very infrequent. And then they become more and more frequent over a rapid period of time, so that soon, without any intervention, these mice or rats will have seizures around 20 to 30 times per hour. And so we were really curious, about what type of brain plasticity is happening in that period of seizure progression, and whether myelin plasticity might play any role in sort of making those seizures become so entrenched in the brain. And so what we did is we looked at myelin structure before and after that period of seizure progression. And what we found is that in two animal models that we looked at, a rat model and the mouse model that have these spontaneous seizures, there was evidence of myelin plasticity and new myelin formation specific to the seizure network after seizures were well established, whereas myelin structure was identical before the seizures started. So we had found a seizure-associated increase in myelin plasticity and myelination. So we then looked at what happens if we prevent the seizures. So if we blocked the seizures with a drug called ethosuximide, which is actually the same drug that we often use to treat children that have this type of epilepsy, blocking the seizures prevented the myelin changes. So that indicated that the seizures are indeed responsible for those myelin changes. So what we think we had found at this point was abnormal activity-dependent myelination in the setting of seizures in a disease model. And the next question was sort of the converse. Okay, if there is seizure-induced myelin plasticity, are those myelin changes having some meaningful impact on how the brain network functions after that? So to investigate the role that myelin plasticity is playing, we decided to block myelin plasticity itself by two different methods. And one was a genetic method where we were able to use knowledge of a specific signaling pathway that's required for myelin plasticity, and we were able to block that through genetic engineering. And in a second approach, we blocked myelin plasticity using a drug that has been previously shown to block myelin plasticity in other work. In both cases, what we found is that blocking myelin plasticity didn't get rid of seizures altogether, but what it did do is it blunted significantly that period of seizure progression. So it prevented the progressive increase in seizure burden that we would typically see. And this makes a lot of sense, actually, because the animal models that we were using still have the genetic change that caused the seizures in the first place. But what they were lacking was this ability to undergo activity-dependent myelination that it seems sort of compounds the pattern of seizures over time and reinforces that seizure pattern. So to wrap it all up, what does this all mean? We think that seizures are inducing myelin plasticity and myelin formation and myelin remodeling that in turn reinforces the seizure pattern and contributes to epilepsy progression. And this was the first study to investigate these questions and to demonstrate that. And so, for some forms of epilepsy, this may open up a new area of investigation and a new, sort of avenue for potential therapeutic approaches. So I definitely, don't want to claim that this same mechanism is at play in all forms of epilepsy, because different epilepsies, have very different mechanisms, and they occur at different times of life and in different regions of the brain. So one of the tasks that my newly formed research group at Stanford is taking on is looking at different types of epilepsy. And one of the first types of epilepsy that we're looking at right now is Lennox-Gastaut syndrome, which is developmental and epileptic encephalopathy and a severe form of pediatric epilepsy without a good, definitive treatment. One reason that we chose that form of epilepsy to look at next is it has some overlap with the mouse models that we already looked at. We were looking at generalized epilepsy with absence seizures, and Lennox-Gastaut syndrome involves some other types of generalized seizures that involve the same network of the brain, including atypical absence seizures. But I think that it will be fascinating to sort of define the myelination phenotype in different types of epilepsy, such as temporal lobe epilepsy, for example, which is a very different type of focal epilepsy. And we may find different things in different forms of epilepsy. So I think it's a really interesting question. And, we have a sort of a whole new area of investigation.